I want to talk to you today about viruses and whether they exist or not. Now, I've written about this before and done a few videos, but this one's probably going to get me into the most trouble due to differing opinions with people that I know. And that's probably a good thing because humanity never really advances until we openly debate and question the nature of things. There is confusion among the health freedom movement over the existence of viruses. Most of the confusion arises from the terrain theory versus the germ theory. So let's look at the germ theory first. So the germ theory has four key concepts. The first of which states that an organism called a pathogen is the cause of disease symptoms. Now, this pathogen can be a large parasite like you see here, or fungus visible to the naked eye, or a smaller bacterium, or a protozoan that's visible to conventional light microscopes, or an extremely small virus visible only to electron microscopes. Now, these are supposedly influenza viruses that were grown in canine kidney cells and spun down with a centrifuge to purify the sample. Now, this image is not completely pure because some cellular debris remained that you can see right here. Now, the second key concept is that the pathogen can transmit between humans, between animals and humans, and from surfaces or food or water to humans. It can be direct contact, like you see here, or it can be droplets or surfaces, and these are called fomites, or it can be food, water, insects, or animals, and these are all called vectors. The third key concept is that the pathogen can multiply within the human, and this leads to disease symptoms. What you see here is the bacteria called E. coli multiplying viewed with a conventional light microscope. And the fourth concept is the disease can be controlled by targeting the pathogen. Now, there are some partial truths in these four key concepts. So let's look at each one. A German physician and microbiologist named Robert Koch developed Koch's postulates in 1884 that are used to prove the first key point, that a specific disease is caused by a specific pathogen. Now, Koch's postulates also have four key concepts. So let's look at those one by one also. First, the pathogen must be present in all cases of the disease. Now, that statement is false. Over the years, we have found that the pathogen can be present in people who do not show disease symptoms. A classic example is the Streptococcus bacteria that most of us have in our throats right now, yet we do not display symptoms of strep throat. Second, you must be able to take the pathogen out of the sick person and grow it in a cell culture. Now, this is done all the time. The problem is you can also grow pathogens from people who are not sick. And you may have even done this yourself in science class by swabbing your mouth and watching nasty stuff grow in a petri dish. The problem is, and virus deniers are very quick to point this out, is that you can grow bacteria in a petri dish because bacteria are living things. But you cannot grow viruses in a petri dish because viruses are not alive and they need living cells to be reproduced. Third, you must take the pathogen you just grew and expose it to a healthy, susceptible person without symptoms and create the disease symptoms in that person. Now, this has also been done, but it does not work every time. The person or animal must be susceptible, and that's a significant problem for proponents of the germ theory. The most recent example of this was done in 2020 by challenging 36 healthy young adults to the SARS-CoV-2 virus. They all got the same exposure, but only half of them got symptoms. And you can read that study right here. Fourth, you must be able to isolate the pathogen from the newly infected person and show that it is the same as the one that they were exposed to. Now, this has also been done, but as we found out earlier, you can also isolate the pathogen from people without the symptoms. So Koch's postulates demonstrate clearly that the first key concept of the germ theory is false. Pathogens are involved for sure, but they are not the cause, simply because some people exposed to the pathogens never display symptoms. Epidemiology, the study of how communicable diseases move through the populations, establishes associations, not causations. Now, the second key concept of the germ theory is that the pathogen can transmit. And we know that is true. The PCR test proved that. Millions of people tested positive for the presence of the virus, but they never got sick. In fact, they could test positive one day, negative the next, and positive again a few days later. And some of these positive tests were false due to excess cycling of the machine, but some were true positives. And this shows us that pathogens move between humans all the time. And they do. In fact, it is estimated that we all inhale about 100 million various viruses every day. Some are associated with disease, others are not. Nevertheless, this transmission is an inescapable fact and cannot be stopped any more than we can stop the ocean tides. The third concept is that the pathogen can multiply within the human, and this will lead to disease symptoms. Now, part of that is true, but not all of it. In fact, if you look at polio, you will see that 70% polio infections, where the polio virus has been found inside the infected person after it multiplied, are completely asymptomatic. You can read that right here on the CDC website. The fourth concept is that the disease can be controlled by targeting the pathogen. Well, that's obviously true, but the problem is when you start trying to kill them off, they can become stronger and more resistant. So it's more effective and safer to control the vector than the actual pathogen. So due to all of these questions and problems, along comes the terrain theory to challenge the germ theory. Now, while the germ theory proposes that diseases are primarily caused by microorganisms such as bacteria and viruses, the terrain theory suggests that it's the internal environment or the terrain of the body that plays a more significant role in determining health and disease outcomes. The terrain theory emphasizes the importance of the body's internal environment, including factors like pH, toxicity, nutritional status, and overall health, in influencing disease susceptibility. It suggests that maintaining a healthy internal terrain can protect against these outward illnesses. The terrain theory recognizes that individuals have varying levels of susceptibility to disease based on their unique internal terrains. It suggests that factors like genetics and lifestyle choices can contribute to these differences. Proponents of the terrain theory often advocate for the preventive measures that support and strengthen the body's internal terrain, such as proper nutrition, detoxification, and stress reduction. While the germ theory attributes diseases primarily to specific microorganisms, the terrain theory acknowledges the presence of microorganisms, but suggests that they are opportunistic and become pathogenic only when the internal terrain is compromised. In this view, microorganisms are seen as secondary contributors to disease rather than the primary cause. The terrain theory highlights the role of maintaining balance and homeostasis within the body as crucial for overall health. Balancing the diet, lifestyle, stress, and environmental exposures are believed to keep the terrain in homeostasis with external factors like bacteria and viruses to maintain health, and that health is not always the absence of symptoms. Comparing the two theories, the terrain theory seems to be more valid. The terrain theory recognizes that microorganisms are always associated with a disease process, but they are not the ultimate cause, for if they were, everyone who came in contact with them would have the disease. But somehow, some proponents of the terrain theory take it a step further and claim that viruses do not even exist. And here's how that came about. In order to prove that viruses exist, three things need to happen. They must be purified, isolated, and then viewed with an electron microscope separate from everything else. Now, this has actually been done many times. Virus deniers will even admit that it was done with a certain type of virus called a bacteriophage that is specific to bacteria, not human cells. What they do is they purify bacteriophages by filtering out the large bacterial cells first, and then they use a centrifuge to separate the remaining particles out by density into these layers that are absolutely pure. Once that's done, they pull off the layers that do not contain bacteriophages so that nothing is left but bacteriophages. That's called isolation, and you can see that being done right here. Then they are viewed with an electron microscope. Now, bacteriophages have a very unique shape that is not ever seen in human biology. So this is probably why virus deniers will admit that the bacteriophage virus does exist, because we clearly see the unique shape that is seen here. There is no way to refute this. It has been done millions of times. Scientists have even taken this a step further and tagged the bacteriophages with fluorescent molecules that will illuminate bright green and then can be seen with a conventional light microscope. Here we see the bacterium making copies of the bacteriophages until it bursts apart and is destroyed. Notice that the bacteria that are not infected are growing, but the infected ones don't get any bigger. That's because the bacteria is using all of its resources to make more bacteriophages, not more of itself. It's important to know that bacteriophages and viruses are not living things. They are not capable of making copies of themselves. The truth is, living bacteria make copies of the bacteriophages, and living cells will make copies of viruses. You might be wondering if this fluorescent tagging has ever been done to particles that are supposedly viruses and filmed them being taken in by cells. Well, the answer is yes. This is the replication of HIV in epithelial cells that was published in May of 2018 by a team of French researchers. Here is the link to the publication and the videos. You may also be wondering how they do this fluorescent tagging. Okay, we got to get into some deep science now. Every living organism, including you, is made of proteins and fats. Now, the message to make those proteins is in your DNA, that is in the nucleus of your cells. So when proteins need to be made, an enzyme called RNA polymerase separates your double-stranded DNA into a single strand that is used to make this messenger RNA that you see happening here in a process that's called transcription. The messenger RNA then migrates from the nucleus of the cell into the cytoplasm, and that's what you see happening here. The messenger RNA is then used by structures in your cells called ribosomes to actually make the proteins. So messenger RNA is a string of nucleotides, of which there are only 4: adenine, uracil, guanine, and cytosine, abbreviated AUGC. Now, the order of these letters is the genetic sequence and will determine what protein will be made. So the single strand of your DNA is the template for making this messenger RNA. And the messenger RNA then becomes the template for making a protein. Now, the transfer RNA that are in the cytoplasm of your cells are what are going to bring in the 20 different amino acids that are going to chain together to actually make the proteins. Each transfer RNA has 3 nucleotides that will bind to 3 nucleotides that are on the messenger RNA. These are called codons because every 3 nucleotides codes for a certain amino acid. Now, in this process of making proteins, A will only bind to U and G will only bind to C. The ribosome chains the amino acids together. The transfer RNA falls off and gets used again. The messenger RNA template is also broken down into individual nucleotides so that they can be used again also. This messenger RNA is the template for what is going to be used to make the protein in a process called translation that you saw happening there. The transfer RNA will bring in the amino acids, which are the building blocks of proteins, and the amino acids get chained together. So scientists have learned how to chemically modify the order of the letters to have those ribosomes make whatever protein they want. And that should concern you because that's exactly what mRNA shots do to you. They're telling the ribosomes of your cells to make a different protein that is not you. So, what they do with the message that is in the genome of the virus to make virus proteins is they modify it so that a fluorescent protein will be part of the protein shell of the virus. Then you can see them when enough of them are present. That's just one way they do it. There are other ways. By modifying fluorescent dyes to attach to viral proteins without actually modifying the genetic code of the virus. So here's the problem that's pointed out by virus deniers. How do you know that what got tagged is really a virus and not something else that's moving between cells and entering cells, which happens in biology all of the time? Since electron microscopy is only a still image, how do you know if this thing that looks like a virus is leaving the cell through a process called budding or entering the cell through a process called endocytosis? There's no way to tell. Also, little things called exosomes that can look exactly like viruses and are similar in size and shape can leave the cell, be taken up by cells, and move between cells, just like viruses do. There can also be cellular fragments that look like viruses, but they're not viruses. There is a big difference between them, though, and this is very important. The difference is exosomes do not cause antibody production because they are made of self, but virus particles do result in antibody production because they are not self. Viruses are made of foreign proteins that result in antibody production that can be detected and measured. Exosomes, although they can look exactly like viruses, do not result in antibody production. So, looking for the presence of antibodies is a way to tell the difference. But that is not going to be good enough for virus deniers. So we can take it a step further to show that the particles are not exosomes. We can do what is called genetic sequencing. Now, here's how that works. We do exactly what was done before by filtering and centrifuging to purify the layers based on density. And after the layer is isolated, they take it a step further by dissolving the protein coat of the bacteriophages or the viruses with enzymes called proteases. Then you purify the layers again with centrifugation, and you isolate the layer that contains only the genetic material. The genetic material that remains is the complete genome of the virus, a long continuous strands of letters, if you will. It's not all chopped up. And this is proven by a process called electrophoresis. The smaller strands of genetic material are going to migrate out here, but the longer strands stay down here. This proves that they have long strands, not little chopped up pieces. Then they get the sequence of the long strands by a highly accurate and repeatable procedure that's called Sanger sequencing. Here's a basic description of Sanger sequencing. It is known that cytosine and guanine always bind together, and thymine that's in DNA, or it would be uracil in RNA, will only bind with adenine. So they add those individual nucleotides to the substrate that contains this unknown long continuous strand of letters. And then they heat up the mixture so that the nucleotides will start binding to the string. And this is called the extension process. But here's the secret. They purposely modify some of the nucleotides so that the extension process will immediately stop when the modified nucleotide is bound, chopping off the long string into shorter strings. Also, the modified nucleotides have fluorophores added to them that will light up a different color based on which nucleotide lights up when it was hit with a laser. Then they run these small strings through a process called electrophoresis that can determine the lengths of strings by one nucleotide difference. This allows the scientists to determine the order of the letters with a high degree of accuracy, and it is repeatable between different laboratories that are doing the investigations. In other words, you can give different labs the same unknown genetic strand. They can sequence it and come up with the results that are almost exactly the same without consulting each other. This is real repeatable science that virus deniers have a hard time explaining. The differences between laboratories that sequence unknown genetic strands without telling them what it is less than one nucleotide in a strand of 1,000. Virus deniers want you to believe that the genetic sequences are just made up by sophisticated computer software that strings together small genetic sequences into a large one that is unique from its source, like this human liver cell that you see here, and then they call that a viral genome. They say it's all just made up, and the sequence does not really exist. So here's how that misinformation got started. Decades ago, when we didn't have the sophisticated detection mechanisms that we use today, cell cultures were used to get enough particles we call viruses to look at. You need living cell cultures to get more viruses because the cells use their own resources of nucleotides and amino acids to make more viruses. Again, it is the living cell that is making more viruses, just like the living bacteria makes more bacteriophages. The virus is not making copies of itself, as it has no intelligence to do that. The living intelligence within the cell is copying the virus. And the message to make the virus is within the virus. The nucleotides, the transfer RNA, the amino acids make the proteins that make more viruses. And this is all coming from within the cell. This depletes the cell resources, sometimes to the point of cell death. But virus deniers are quick to correctly point out that cells in cell cultures will always die, whether materials that contain viruses were added to them or not. And when they die, cell fragments can appear that look exactly what viruses are supposed to look like. And you can see them here. All of this is true. This is a scanning electron microscope image of a cell dying through normal death, and this is called apoptosis. Here is the link where you can see the image and read more about it. Also, when cells die. Their genetic material ends up getting fragmented into smaller strands. Here is a transmission electron microscopy of a strand of genetic material. Now, these strands after cell death are generally about 100 to 200 base pairs long. Virus deniers admit that these smaller strands can be sequenced, and this is done all the time by the Sanger method. Then they say that these smaller genetic strands that were sequenced and verified can be assembled with sophisticated computer software to come up with a made-up, long sequence that is thousands of letters long, unique and different from the cellular genome that it originally came from, and then record that as a made-up viral genome. Now, you can certainly have a computer randomize these short strands into something very long and unique, but I propose that you will not be able to end up matching a verified genetic sequence of a virus. So here's an experiment that needs to be done. You take the same cell culture and you separate it into two groups, one of which you will add a substrate purported to contain viruses, and the other you will not. You treat both these groups the same as far as nutrients, antibiotics, and chemicals to be added. Both cultures will experience cell death, either by necrosis due to viral infection or apoptosis due to starvation or chemicals. Either way, you will end up with cellular fragments. This part of the experiment has already been done, but we need to go further. In both groups, we need to use ultracentrifugation to purify all of the particles into different layered densities. Most likely, you will end up with similar layers in both groups. Then you isolate the layers, and in the layer that purportedly contains viruses, have at least two different labs sequence the long, continuous genetic strand that will be there without consulting each other, and their results should be the same. Next, you take the similar density layer from the culture that the viruses were not added to, and you sequence that as well. The long, continuous sequence that was found in the viral sample should not be there. You will have smaller sequences that resulted from cellular death or apoptosis, and they will be 100 to 200 letters long. Next, have the sophisticated genomic sequencing software try to assemble these fragments into a sequence that will match what came out of the viral sample. My guess is that they will not be able to do it. They will be able to do it if they would use enzymes called nucleases to break these genetic fragments into smaller pieces that are just a few nucleotides long, but that would be cheating. Now, to my knowledge, this experiment has never been done. If it has, I would like to see it. Another major complaint of virus deniers is that cell cultures are used, and that viruses should be able to be purified and isolated directly from clinical samples without that cell culture step. Viruses have been imaged directly from clinical samples using various techniques since the early 1980s. And here is one study from 1981. But virus deniers will still ask, how do you know what you're looking at is really a virus? Well, consider this very recent study where antibodies against influenza viruses were coated with magnetic nanobeads. Antibodies will not attach to exosomes or other cellular fragments, but they will attach to viruses. The magnetism aggregated the virus particles together directly from a clinical sample, not a cell culture, and then they were viewed by electron microscopy. You can read it right here. My opinion is that viruses do exist in the form of minuscule transmissible particles that are not alive. They have no intelligence and no desire to attack you. So what good are they then? What is their purpose for existence? Well, here's my theory, and it's not new by any means. Doctors and scientists have been considering this for a very long time. Life is about survival. And in order for man to survive the poisons and toxins and radiation that we keep insisting on putting into the air and the food and the water, we need to adapt cellular functions to tolerate these things, or we would die off as a species. So to change cellular functions, we need to make new proteins. So where could the message to make these new proteins be coming from? You were born with the DNA from your parents, and in that DNA is the message to code the proteins that make you what and who you are. The message is transcribed into messenger RNA here and then translated into proteins here. But if you need to make a new protein to change cellular function in a particular cell line, the message to make that protein has to come from somewhere else besides your DNA. Now, admittedly, there is editing of the genetic sequence of the natural messenger RNA that happens all the time due to little segments of genes that are called transposons that move around and jump around, and also due to the actions of ApoBec enzymes that are in your cellular cytoplasm. But these changes are going to be minor. If you need a really big change, the message must come from outside the body. It's very possible that these messages could be contained in transmissible virus particles that reverse transcribe into our DNA that will ultimately result in new protein production that help us adapt to the environmental changes that we ourselves created. And the fact that human cells have receptors that will only attach to certain viruses means that we were meant to communicate with these things. Viruses could actually be an interspecies communication mechanism to keep our genomes updated. And when man tries to block this communication from happening by using drugs or shots called vaccines, the cells of our bodies will mutate the virus so that it can continue to transmit. Always remember that it is us that mutates the virus. It is not the virus mutating itself. It is not alive, and it doesn't have the intelligence nor the desire to do that. When science and organized medicine finally recognizes that viruses are a necessary part of the ecosystem that we should be coming into harmony with, instead of trying to destroy them, the way we look at communicable diseases will change forever. If you want to learn more about the immune system, consider taking my comprehensive 4-module course available here. Thanks for watching, and thanks for staying smart.